Good morning guys, welcome to another Biology with Miss Cherry. Today we are looking at a not too difficult but also not too easy idea and concept of epistasis. So to begin with, have a go at these questions. We've got a flowchart at the bottom showing the transition between a brown pigment to a red pigment to a blue pigment that are controlled by enzymes A and enzymes B. Okay? Dominant alleles produce functioning enzymes. Now, what happens to the pathway if an organism has the genotype AA and then something else after it? And then what happens to the organism if the pathway is something and then two little b's? Okay, so usually, you can solve this problem. It's not too difficult. It'll get a little bit more difficult later. But for now, just have a think, have a go, pause, spend a few minutes, and then come back when you're ready. All right, so as we said, today we are looking at epistasis. All we've got to be able to do is describe it and recognise it happening in the transition of organism phenotypes or the variation in organism phenotypes. So the starter showed an example of an epistatic pathway or a pathway where epistasis is in action. Now, can you have a look at the example again? It's exactly the same one as the starter at the top. We've got brown going to red, going to blue phenotypes. Controlled by enzyme A and enzyme B, and have a go at defining what epistasis is. Now, definition-wise, it's quite a key one to be able to regurgitate because it's often one of the ones that has underlined words in the answer, which means that you have to be able to get the key terminology correct. So epistasis is when the allele, so the variant of one gene, so maybe big A or little a, affects or masks the expression of another gene in the phenotype. So we're looking at a combination effect here. And we're looking at more than one gene. You're not going to get epistasis in a question that's only monogenic. There's something to bear in mind. And the example, for some reason, that they want you to use in the exam is looking at mice fur and their colouring normally. So let's get started. Let's have a look. So this is the standard example, as we've said, it's mice for your A levels. And we've got two genes controlling the colouring of that fur. So gene A controls the distribution of black pigment or melanin in the mice fur. And if we've got a big A, if I draw a fur here and a fur there, if you've got a big A genotype, then you end up with some banding shapes. And if you've got a homozygous recessive or a recessive A allele, then you've got all black hairs. Gene B, on the other hand, controls the production of melanin. Okay, so A is for the distribution of the pigment. Gene B controls the production of the melanin. Okay, so gene B is actually there to control gene A. And if we've got big B, then we're producing melanin, we're getting that pigment. But if we've got a little b, then there's no melanin or no pigment. And that means that we're not going to get any coloration. So thinking about it logically, have a look at these three phenotypes, 1, 2, 3 at the bottom, and have a go at figuring out what the mice fur will look like. Should take you a minute or two, so pause and play again when you're ready. Alright, so let's take a look at each of these in turn. Now if you remember, the presence of B, or big B, gives a pigment, and little b is no pigment. A, or big A, is banded, and little a is fully black. Now, also in the previous slide, we saw that B controls A. So firstly, what we can do is look at each of our genotypes and decide whether the pigment is going to be produced. So for this first one, I know I'm going to get a pigment. For the second one, I know I'm going to get a pigment. But for the last one, big A, little b, there's no pigment. Okay, that means that we've got two of those recessive alleles, no pigment produced at all. And only then can we start taking a look at the A alleles, because we know that B controls A, so it's probably better, logically thinking wise, to look at B first. Now for the first one, we've got two big A's, two of those dominant alleles, which means our phenotype is going to be black banded hairs. We know we've got that pigment and we've got the banding available to do that as well. And these black banded hairs are called agouti, A-G-O-U-T-I, just there. Okay, and if you wanted to give that reason, it's got the big banding or the big dominant allele A for banding and the melanin producing big B. Conversely, we've got the pigment being produced in the second example, but there's no dominant banding allele. So instead we just get uniform black hairs. The final example though is maybe a little bit more confusing, but hopefully you will have got there because I'm sure you're all plenty capable. So we've got two little b's. We know we're not producing any pigment. And that sort of makes the presence or absence of a dominant A allele irrelevant, really, because all we're getting is a lack of pigment, which means that our phenotype is albino. Now, regardless of this dominant A allele, as we've said, there's no melanin producing allele in this genotype. There's no big B or big B, little b, so we get no colour regardless of whether we've got that big A or a little a. Now, could you please use your knowledge, okay, so thinking about things a little bit deeper, what is the phenotypic ratio produced by the following? A homozygous agouti mouse, so remember that is a banded mouse, crossed with a homozygous albino mouse. And remember that's no pigment. Okay, have a go. Pause, you're going to need to do a genetic diagram, a big cross, and then come back when you're ready. So we know we're starting with a homozygous banded organism, so we know that we're going to have two big A's and two big B's, because I said they were homozygous, and then a homozygous albino mouse as well. Now, gametes-wise, regardless of whether I go 1, 3, 1, 4, 2, 3, 2, 4, I'm just going to end up with big A, big B for parent 1, and little a, little b for parent 2. So I can put that into a really nice simple genetic cross, which looks a little bit like this, and you should have seen that all of the offspring have the agouti dominant phenotype. This is not the epistatic phenotype. Okay, the epistatic phenotype is the phenotype produced by the presence of an epistatic gene, and in this case it's B that is controlling the A, and that's your epistatic gene. The phenotype produced by presence of epistatic gene. Okay, and now we're going to think about that in a little bit more detail, because I would like you to have a go at crossing two of those F1 generations. Okay, what are the offspring of two of those offspring going to look like? What is the genotype and the phenotypic ratio? Pause again and play when you're ready. So for this example, we're crossing two parents that are heterozygous for both of those genes. And as you can see, we're going to be using the 1, 3, 1, 4, 2, 3, 2, 4 examples. So 1, 3 is big A, big B, 1, 4, big A, little b, 2, 3, little a, big B, and 2, 4, little a, little b. Both of those parents have those potential gametes, which means that we are going to end up with a 4x4 Punnett square, and your offspring ratio, your phenotypic ratio, is 9 agouti to 3 black to 4 albino. Okay, now we've got to get into some terminology now, and that is that this is an example of recessive epistasis. And we say it's recessive epistasis because we need 2 copies of that epistatic gene. Remember we said that B is controlling A, and therefore B is the epistatic gene. And the production of those 2 recessive epistatic alleles means that regardless of what A is, it's going to mask that phenotype. Okay, so as you can see in, if I circle this offspring, those 2 little b's mean that regardless of the fact that realistically those bits of fur, those fur strands, hair strands, should be banded because they've got those two recessive B alleles that are controlling the production of that banding pigment, they're not going to be there. Okay, it's commonsensical, but we need to know the language surrounding it as well. So this is the first rule. Okay, in recessive epistasis, the idea that two recessive epistatic alleles, so two little b's in this example, from a parental cross of a homozygous dominant with a homozygous recessive parent, so in this case we did big A, big B, times little a, little b, that gives an F1 offspring, so the first group of offspring that are 100% heterozygous, or in this case, big A, big B, little b. And then if we cross those again, so if we cross F1 times the F1, then we end up with a 9:4:1 ratio of dominant both, i.e. big A, big B, to 4 dominant epistatic recessive other. Now that means that our epistatic alleles are going to be dominant, so we know that we're looking at big B something, so that could either be 2 big B's or big B, little b. And recessive other means that we're looking at 2 of our recessive A alleles, the ones that are controlled by the presence or absence of that epistatic gene. And then finally, 1 recessive epistatic. So that could be something, and we have to have 2 of those recessive epistatic alleles, the B's that are controlling the A. And that, in this mice example, gave us the albino phenotype. Now here we've got another example. Squashes, like the vegetable, like butternut squash, are also a lovely example that they like to use in the exam. And squashes have got colours controlled by a couple of genes as well. So we've got the epistatic colour gene, big W or little w, and the yellow gene, big Y or little y. Now what this means again is that the presence or absence of W is controlling the Y. Okay, so in squashes, the no colour or big W pigment is dominant over the colour pigment, which is a little w. Okay, so no colour is big W, colour is little w. And then yellow, big Y, is dominant over green colour, little y. So have a go at figuring out what are the possible genotypes of a white squash. Look at the information above, pause the video now, and have a go at figuring out. You will need 6 different answers. Okay, there are 6 possible combinations of genotype for a white squash. Think carefully about them, and then play again when you're ready. Okay, so for white squash, we know that we're going to need at least 1 big W. Okay, because remember that big W is for no colour and it's controlling the yellow or the green. Okay, so you could have had all of these. I'm not going to read them all out, but notice that we've got 3 homozygous dominant for the no colour and also 3 heterozygous for the no colour. So the presence of only 1 big W allele is needed to make the squash white. Now again, I've sort of not followed my own instructions here because this example has got letters that look very similar, upper and lowercase. This is an example from the exam board though, so don't blame me, don't shoot the messenger. But let's take a look at how it goes in a Punnett square. So a homozygous dominant squash is crossed with a homozygous recessive squash. Could you please use the key and then answer the questions below? So what are the colours of both parents, a homozygous dominant squash and a homozygous recessive squash? And then what is the phenotypic ratio of the offspring of the cross? So this should take you a couple of minutes. Pause now and play again when you're ready. So we're looking at a homozygous dominant parent, so big W, big Y, and a homozygous recessive parent, so little w, little y. Now both of those crossed together, we're going to get the gametes big W, big Y for this parent and little w, little y for the other parent. And your Punnett square should look something like that. Okay, so parent 1 over here, the 2 homozygous dominant ones, is white, because remember those big W's control the presence or absence of colour, whereas this parent over here on the right hand side is green, because they've got colour but they're recessive for the colour allele as well. So they're not yellow, they're in fact green. And all of our offspring, our F1 generation, are 100% white. Okay, they are 100% epistatic because that big W is masking the Y allele. And then have another go, as we did last time. What happens if we've got two of the offspring from this cross? So I'll highlight those for you. Any of these two cross together. So pause again, do me a nice big Punnett square. Tell me the F2 phenotypic ratio. Pause and play again when you're ready. So again, we've got a nice example, same as we did last time, where we've got a heterozygous parent, or two heterozygous parents really, and we're using our 1, 3, 1, 4, 2, 3, 2, 4 rule. So our parent gametes are big W, big Y, big W, little W, big Y, little w, little y. Both of the parents are that, so we end up with the following cross. And you should have recognised that we have got 12 colourless, 3 yellow and 1 green. Hopefully you spent some time going through and recognising that if there was a big W in any of those boxes, then they were going to be colourless or white. And this is an example of something called dominant epistasis. So we had a look previously at recessive epistasis, when the presence of two little b alleles stopped the mouse being coloured. It just stayed as an albino mouse. Whereas this example, dominant epistasis, is where one or more copies of the dominant epistatic gene, so in this case big W, masks the expression of the other gene. Okay, so as we said, in all of these offspring, regardless of whether they've got two big W's or a big W and a little w, that big W is basically going, nah, no colour, you're going to be a white squash. Dominant epistasis, that dominant allele stops or controls the phenotype. And this is, as we've said, dominant epistasis. This is the rule, so as per when we looked at the rule for recessive epistasis, if we've got a homozygous dominant and a homozygous recessive parent crossing together, then the F1 genotype is going to be 100% heterozygous, and generally this is the epistatic phenotype. So in this case it was a white squash. And then for the F2 generation, we should always get a 12:3:1 ratio of the dominant epistatic. So in this case it's a presence of a big W and then something else. And then 3 recessive epistatic dominant others. So that means that we've got 2 little W's and then a big something. So in this case it was a big Y and then a something else. And then finally recessive both. And this is where we've got 2 recessive W's and 2 recessive Y's. And that gives us our green squash in this example. Okay. Now thinking back to the starter, what happens to the pathway if little a, big B is expressed and Y? And what happens to the pathway if big A, little b is expressed and Y? So to look back to your answers, maybe you need to make some changes, maybe you don't, maybe you still think you're confident in your answer, but have a go. Again, just double check. And this is what your answers should have looked like. So if we've got 2 little a's, okay, and we know that a big A is a functional enzyme, a dominant allele is a functional enzyme, then for this first example, the pigment is going to stay brown. Okay, we don't have enzyme A to catalyse the formation of the red pigment from brown to red, so we don't have any further colour change regardless of the presence of those 2 dominant B alleles. Conversely, for the second example, with 2 big A's and 2 little b's, the pigment is going to stay red. And this is because we've got a functional transition, that enzyme A is functioning properly, so brown can turn into a red pigment, but that red pigment cannot turn into the blue pigment because enzyme B is non-functional. Okay, and again, remember, it was that dominant B allele that made it work. Now, if you're in my class, then please, I'm going to put the summary questions from page 178 in the blue book onto OneNote, and this is from the Oxford Year 2 book.
And then I'll put the answers on as well so you can self-assess. And then we'll have a look at some exam questions afterwards. So pause now and have a go at these 2 tasks for me and then play again when you're ready. All right, so with these epistasis questions, there's normally a lot of words and a lot of gumph that surround the topic itself. So in this example, rats have a dominant allele big B, which gives a black coat colour, and a recessive allele little b, which gives a cream coat colour. This gene acts in association with a randomly assorting epistatic gene, which possesses 2 alleles. Dominant E, or big E, which allows the development of colour, and recessive E, little e, which suppresses colour development, resulting in albino rats which have white coats and pink eyes. Write down the possible genotypes of the following rat phenotypes: a black rat, a cream rat, and an albino rat. So this is about 3 marks. I'm going to give you should pause for about 4 minutes to think properly about this. So we're looking at a black rat, a cream rat, and an albino rat. Pause and play again when you're ready. For question B, what do you understand by the term randomly assorting epistatic gene? Now, this is 3 marks, so you might want to take each of those terms in separation. So you might want to think about what epistatic gene means, what randomly assorting means, what do they mean when they're combined together. You're going to have to think back to some other lessons as well. Random assortment and things like that. We have covered this module, so maybe do a double check, double recap. Pause, spend about 5 minutes on this, full prose for your answer, and play again when you're ready. And the final question, I've put the stem back into the top of this screenshot. It's not a new stem, it's exactly the same. With the aid of a genetic diagram, show the genotypes and phenotypes which could result from a cross between a cream individual of genotype little b, big E, little e, and a black rat heterozygous for both characters. Indicate the ratio of phenotypes possible. And it's 4 marks for a Punnett square. So give yourself maybe 6 minutes, do it properly, lay it out neatly. You can ask if you've got any queries, and then play again when you are ready. So answers-wise, we're going to take these in turn. So for the first question, it was about black mice. So you should have had all of the genotypes possible containing at least 1 dominant B allele. Okay, 1 big B at least. Those are your 4 possible combinations. For the cream mouse, we were looking at recessive Bs, but at least 1 dominant E allele. Okay, so you could have had little b, big E, or little b, big E, little e. And then finally, for the albino phenotype, we're looking at 2 recessive E alleles, because remember they are recessive, so we need 2 of them. We need a pair of those recessive alleles for the phenotype to manifest, and any combination of B alleles. But all of those genotypes on each of those rows are required for the mark for each of those offspring phenotypes. Now, the next question was looking at what does it mean by randomly assorting epistatic gene. And there were 3 marks available, so I'm going to take each of those in turn in a line. So an epistatic gene is one that influences or controls the expression of another gene. And randomly assorting means that the gene is on a different chromosome to the gene it's influencing. So maybe if we're looking at this example, the B locus might be on chromosome 12, whereas the E locus might be on chromosome, 20. And therefore, because we've got that random assortment, it means that we've got independent segregation, or the fact that those genes are not linked. Okay, remember we said about gene linkage a few lessons ago, when they're too close together to be separated and you don't therefore get all of the recombinants? Doesn't happen in this case. They're far apart, they can separate with free will. Each of those lines, again, where the semicolon is a separate mark. If you weren't too sure about what randomly assorting means in terms of the inheritance, maybe pause and make a little note of that important bullet point down there, that it behaves in a non-Mendelian manner, or that it tells you that the genes are not linked. And the final question was a Punnett square. So we had 2 parents. It told you the genotypes, and you had to separate out the gametes. That's the first mark. The next mark is then for the cross, or the 2 marks are for the cross. And you notice here that they've written in the phenotypes of each of the offspring in the boxes. Okay, and the final mark is for the ratio 3 black, 2 cream, and 2 albino. If you didn't get any of those right, let me know. Let me know how I can help. I hope you found this useful, and I will see you next time. Bye bye.